Now here we have question 38. Identify the wrong statement with regard to restriction enzymes. Now restriction enzymes, as you know, they are the molecular scissors, one very important tool for biotechnology for developing recombinant DNA. So in this, they cut the strand of DNA at palindromic sites. Of course, those are the restriction sites, and it is this site which would be specifically present. Only then that restriction endonuclease will cleave the phosphodiester bond. They are useful in genetic engineering. Of course, sticky ends can be joined by using DNA ligase. Now this is a different thing altogether. This is not the function of restriction enzyme. So this is an odd one out. And each restriction enzyme functions by inspecting the length of the DNA sequence. So that's right, it will go all along the DNA sequence, and where the restriction site would be present, that would be the recognition site, there it will bind and cause the cleavage. Okay. So in this case, our answer would be option 3. Now from here, let us move on to next question, that is 39. Now in this question, the infectious stage of Plasmodium that enters the human body. Now you know, for Plasmodium, which causes malaria, the vector is female Anopheles mosquito, and when this female Anopheles mosquito, it takes the blood meal, during that time it injects the saliva too, because the saliva has the anticoagulant which will not allow our blood to clot, and the mosquito can easily suck it. But if this mosquito is carrying the infective stage, even that stage is passed on into our body, into our bloodstream by that saliva, right? And the stage present in it is the sporozoite. So in this case, our correct answer is of course the sporozoites, and these sporozoites, once they are injected into our bloodstream, they move on to the liver where they further multiply. So from here we can move on to our next question, that is question number 40th. So in this, meiotic divisions of the secondary oocyte is completed. Now you see, at the time of ovulation, the female gamete, as yet it is not formed into ovum, it is in the form of secondary oocyte which has started meiosis 2, but it is suspended at metaphase 2. Now this would be present in the ampullary part of the fallopian tube, and the rest of the meiosis 2 will be completed only under one condition, when the sperm enters, that is at the time of fertilization. So let us see the various options available with us: at the time of copulation, after zygote formation, at the time of fusion of a sperm with an ovum, and prior to ovulation. Now this is the only one which we can take it as correct, the third one, although ovum will be formed only when the sperm enters, then that secondary oocyte will finally convert into ovum, and this would be a very short phase, because then immediately fertilization is accomplished and this will turn into zygote, right. So our correct answer is option number 3. This is once again a very important concept that we take up in the human reproduction chapter. So from here, let us move on to our next question.